degeneration. Mucous neck cells helps in regeneration. Parietal cells, they helps in secretion of the HCl and intrinsic factors. Enterochromaffin cells, they secrete the histamine, and chief cells secrete the pepsinogen and gastric lipase, and class 1 G cell secrete the gastrin. So the HCl that is secreted, that enters into the canaliculi. Okay, this is the canaliculi, and the hydrogen ion and chloride ion, they are secreted separately. Okay, in the canaliculi there is the formation of HCl. Right? Directly HCl is not secreted from the parietal cell. Okay, directly HCl is not secreted from the parietal cell. The hydrogen is secreted first, then chloride is also secreted, then in the canaliculi, okay, in the canaliculi, the hydrogen and the chloride, they combine together and forms the hydrochloric acid. So HCl, it is secreted by the parietal cells. Okay, HCl is secreted by the parietal cells of the oxyntic gland. Right? Parietal cells of the oxyntic gland. So omeprazole, it inhibits the hydrogen pump. Okay, omeprazole, it inhibits the hydrogen pump, and it helps in prevention of the gastric ulcer. Right? Now let's read about the mechanism of HCl secretion. Okay, this is very, very important. It has come many a times in the exam. Mechanism of HCl secretion, it is very, very important. Right? So so this is the lumen of the stomach, and this is the interstitial fluid, and this is the parietal cell. Okay, this is the parietal cell. So in the parietal cells there are water. Okay, the water, they are dissociated. The water is dissociated into hydrogen and hydroxide ion. Okay, water is dissociated into hydrogen and hydroxide ion. The hydroxide ion combines with carbon dioxide and forms the bicarbonate. Okay, so this water, when this water is dissociated into hydrogen and hydroxide ion, this hydrogen ion, it is exchanged with the potassium. Okay, this is the primary active transport. This is the primary active transport. Hydrogen ion is actively transported into the lumen of the stomach by the primary active transport, and the pump that is working is hydrogen potassium ATPase pump. Okay, hydrogen potassium ATPase pump actively pumps the hydrogen ion. So when hydrogen ion goes outside, okay, when hydrogen ion goes outside, this hydrogen ion, it is the positive ion, so there must be electrochemical balance. Okay, hydrogen ion is the positive ion, so there must be electrochemical balance. So to fulfill that electrochemical balance, chloride ion must go to the lumen of the stomach. Okay, chloride ion must go to the canaliculi, then to the lumen of the stomach. Okay, so it goes to the canaliculi by the chloride transporter. Okay, it is the passive, chloride ion, it goes to the canaliculi by the electrochemical gradient. Okay, chloride ion goes to the canaliculi through the electrochemical gradient, and this chloride ion is exchanged through the chloride bicarbonate exchanger. Okay, chloride ion, it is exchanged to the chloride bicarbonate exchanger. Okay, so chloride ion, chloride from the interstitial fluid, it goes to the parietal cells, then from the parietal cells it passively goes to the canaliculi of the parietal cells, right? And this bicarbonate ion, it goes to the interstitial fluid, then it goes to the blood. Okay, so after consumption of meal, the alkalinity of the blood, pH of the blood is increased due to the movement of this bicarbonate ion. Okay, due to the movement of this bicarbonate ion, and it is known as postprandial alkaline tide. Okay, the pH of the blood becomes alkaline. Right, pH of the blood becomes alkaline. It is known as postprandial alkaline tide. Let's again see this figure. Okay, let's again see this figure. So this is the lumen of the gland. Okay, this is the lumen of the gland. This is the interstitial fluid. Okay, and this one is the parietal cell. Okay, this one is the parietal cell. So in the parietal cell, water is dissociated into hydrogen and bicarbonate, hydrogen and hydroxide ion. Hydrogen ion, it is actively transported. Okay, hydrogen ion, it is actively transported into the lumen of the parietal cell. Then for working of this hydrogen potassium ATPase, okay, for working of this hydrogen potassium ATPase, there must be always working of the sodium potassium ATPase on the basolateral side. Okay, on the basolateral side, sodium potassium ATPase, this one on the basolateral side, sodium potassium ATPase must always be working. Okay, it must always be working. If sodium potassium pump is not working, then this mechanism will not work. Okay, this mechanism will not work. So sodium potassium pump must always work. So when sodium potassium pump is working, sodium ion is pumped outside, potassium ion goes inside. Potassium ion also goes passively to the lumen. Okay, potassium ion goes passively to the lumen of the parietal cell. Then the hydrogen potassium ATPase, it works and it forms the hydrogen ion actively to the lumen of the gland. Okay, right? So this bicarbonate ion, the hydroxide ion that is formed, it combines with water and forms the bicarbonate. Bicarbonate, it gets exchanged with the chloride. Okay, chloride bicarbonate exchanger. So this bicarbonate ion, it goes to the interstitial fluid. Okay, bicarbonate ion goes to the interstitial fluid and chloride ion comes inside. Chloride ion, it is passively diffused. Okay, it is passively diffused to the lumen of the parietal cell, and in the lumen of the parietal cell, the hydrogen and chloride, they combine together and forms the hydrochloric acid. Okay, am I clear about the mechanism? Am I clear?
Yes, sir.
In the parietal cell, there is the water is dissociated into hydrogen and hydrogen and hydroxide ion. Okay, hydrogen ion, it is actively pumped with the exchange of potassium. Okay, by the hydrogen potassium ATPase pump, then hydrogen ion goes to the lumen of the parietal cell. Then the hydroxide ion that is formed, that combines with water, that combines with the carbon dioxide, because during the metabolic process, when the metabolic processes are going on, there is the formation of carbon dioxide also. So carbon dioxide and hydroxide ion, they combine together and form the carbonic acid. Okay, first they form the carbonic acid, then they form the bicarbonate. So once bicarbonate is formed, bicarbonate is exchanged with the chloride through the chloride bicarbonate exchanger. Okay, chloride bicarbonate exchanger. Then bicarbonate ion goes to the interstitial fluid, then ultimately to the blood side, and chloride ion, it passively diffuses to the chloride channel. So this is the passive diffusion. This is the this chloride ion diffuse to the lumen of the parietal cell through the electrochemical gradient. Okay, through the electrochemical gradient, to balance the electrochemical gradient, this chloride ion moves, because more of the hydrogen ion is being secreted into the lumen of the gland. So to balance the charge, this chloride ion passively diffused through the electrochemical gradient. Right? This chloride ion passively diffused through the electrochemical gradient. The bicarbonate ion that is formed, the bicarbonate ion that goes to the blood side, it makes the blood alkaline. It makes the blood alkaline. So this is known as postprandial alkaline tide. After consumption of the milk, there is the exchange of chloride with bicarbonate. Bicarbonate ion goes to the blood and it causes the, it increases the alkalinity of the blood. This is known as postprandial alkaline tide. Okay, this is asked in the exam also. What is postprandial alkaline tide? You have to remember this figure. Okay, just remember this figure. Water, it is dissociated into the hydrogen and hydrogen and hydroxide ion. Okay, water is dissociated into hydrogen and hydroxide ion. Hydrogen ion, it is um, actively pumped to the lumen of the parietal cell by the hydrogen potassium ATPase pump. Okay, this is the active transport, hydrogen potassium ATPase pump. Hydrogen goes to the lumen. So when hydrogen is formed, okay, when hydrogen is formed in the lumen, to balance the hydrogen, to balance the positive charge, negative charge also diffuse along the parietal cell. Okay, negative charge also diffuse along the parietal cell. So this negative charge, it comes by exchange with the chloride and bicarbonate. So this is very, very important, right? This is very, very important. So in the parietal cell, the water, it is dissociated into hydrogen and hydroxide ion. Okay, hydrogen and hydroxide ion. So this hydrogen, it is actively pumped through the hydrogen potassium ATPase. Okay, hydrogen potassium ATPase. So hydrogen is pumped into the lumen. Okay, hydrogen is pumped to the lumen. Then this hydroxide ion, it is combined with water with the help of carbonic anhydrase enzyme. This is converted into the bicarbonate. Bicarbonate, it is exchanged with the chloride. Chloride ion comes to the parietal cell. So to balance the charge, okay, to balance the positive charge that is in the lumen, that is created in the lumen, this negatively charged chloride ion, it goes to the lumen of the parietal cell, and in the lumen of the parietal cell, it forms the hydrochloric acid. Okay, it forms the hydrochloric acid. And this, the bicarbonate ion that goes to the blood side, it causes the blood pH alkaline. Okay, it causes the blood pH alkaline, which is known as postprandial alkaline tide. Okay, postprandial alkaline tide. So the the importance of this hydrogen potassium ATPase pump is that we are targeting the drug here. Okay, some of the drugs we are targeting here, the proton pump inhibitor omeprazole. Okay, omeprazole is targeted here, right? Omeprazole, it inhibits the hydrogen potassium ATPase pump. Okay, omeprazole, it inhibits the hydrogen potassium ATPase pump. So when this hydrogen potassium ATPase pump is not working, then hydrogen ion will not come to the lumen. If hydrogen ion is not coming to the lumen, chloride ion is also not coming to the lumen, and hydrochloric acid is not formed. Okay, HCl is not formed. So omeprazole, it inhibits the hydrogen potassium ATPase pump here. Okay, here. Omeprazole, it inhibits the hydrogen potassium ATPase pump. So when hydrogen potassium ATPase pump is inhibited, there is no pumping of hydrogen. Hydrogen is not actively pumped to the lumen. So this is the same mechanism, right? That has been explained. Whatever I have explained, that is right. Parietal cell secrete hydrochloric acid into the lumen of the stomach and concurrently absorb bicarbonate into the bloodstream as follows. Right, water and carbon dioxide and water are combined to form the hydrogen and bicarbonate, which is catalyzed by the carbonic acid anhydrase enzyme. Hydrogen is actively secreted by the hydrogen potassium ATPase pump. Chloride is secreted along with this passively. Okay, that is passively secreted, and this is the drug omeprazole. It inhibits the hydrogen potassium ATPase pump. The bicarbonate, it is exchanged with the chloride, and this is known as alkaline tide, postprandial alkaline tide. This is the resting state. Okay, this is the resting state of the parietal cell, and this is the active state of the parietal cell. Right? You can look at this figure. This is the resting state of the parietal cell, and this is the active state of the parietal cell. So mainly the three mechanisms help in secretion of the parietal cell. Right? Mainly the three mechanisms help in secretion from the parietal cell. One is through the acetylcholine, one is through the histamine, and one is through the gastrin. Okay, acetylcholine, histamine, and gastrin. They are responsible for secretion of hydrochloric acid from the parietal cell. So when the acetylcholine, histamine, and cholecystokinin, they are not acting on the parietal cell. This is the parietal cell. Okay, this one is the parietal cell. So when the acetylcholine, histamine, and cholecystokinin, they are not working on the parietal cell, the tubular vesicles that contains the hydrogen potassium ATPase pump, they are internalized in the cell. Okay, they are internalized in the cell. Once the acetylcholine binds, or the histamine binds, or the gastrin binds, these hydrogen potassium ATPase, they fuse to the lumen of the they fuse to the apical side of the parietal cell. Okay, so this is the active secreting parietal cell, and this is the resting parietal cell. During the resting state, the hydrogen potassium ATPase pumps, they are internalized inside the parietal cell. They are present inside the parietal cell. Once acetylcholine binds, once histamine binds, once gastrin binds, these pumps, they will be taken to the, they will be fused to the apical cells. They will be fused to the cells of the parietal cells. Okay, towards the lumen. Okay, they will be fused towards the luminal side of the parietal cell. Right? Am I clear? Am I clear? So now let's read them. Let's build the mechanism of how acetylcholine, histamine, and gastrin works. Okay, right? Mechanism of how acetylcholine, histamine, and gastrin. So acetylcholine, right? Acetylcholine, it acts on the M3 receptor present in the parietal cell. Okay, acetylcholine, it acts on the M3 receptor present on the parietal cell. Acetylcholine increases the level of the calcium ion. Okay, acetylcholine increases the level of the calcium ion. Acetylcholine and gastrin, it increases the level of the calcium ion. So when calcium ion is increased, what will happen? It will cause exocytosis of the synaptic vesicles. Yes or no? We have studied in the MSK also. How many calcium ion are required for exocytosis of one synaptic vesicle? Four. Okay, so acetylcholine, when it binds with the M3 receptor, it causes the increase in the calcium level of the parietal cell. So once parietal cells calcium is increased, these tubular vesicles that contains the hydrogen potassium ATPase, they will be fusing to the luminal side of the parietal cell, and they will be causing active secretion of the hydrochloric acid. Likewise, gastrin, it acts on the CCK-B receptor, cholecystokinin B receptor. Okay, gastrin, it acts on the cholecystokinin B receptor of the parietal cell. Okay, gastrin, it acts on the cholecystokinin B receptor of the parietal cell, and it also causes the increased level of the calcium ion. So when calcium ion is increased, it will cause exocytosis of the synaptic vesicles, tubular vesicles consisting of hydrogen potassium ATPase pump. Then there is the histamine. Histamine acts on the H2 receptor. Okay, so this H2 have more potent effect than the acetylcholine and gastrin. Okay, if you are just blocking these H2 receptor, then most of the problem will be solved. Okay, most of the problem will be solved. So the antihistaminic drugs, ranitidine, cimetidine, ranitidine, they act on the S2 receptor. Okay, they act on the S2 receptor. When they act on the S2 receptors, they will inhibit the formation of cyclic AMP, and these tubular vesicles will not be fused. Okay, histamine, it acts on the S2 receptor. Okay, histamine, it acts on the S2 receptor. When it acts on the S2 receptor, cyclic AMP level is increased. When cyclic AMP level is increased, then it will cause exocytosis of the, it will cause fusion the lumen of the parietal cell. Likewise, gastrin also stimulates the GQ receptor. Okay, it also stimulates the GQ receptor. Again, it will stimulate the phospholipase C. IP3 is activated. When IP3 is activated, endoplasmic reticulum will secrete the calcium ion, release the calcium ion. Calcium ion, it binds the tubular vesicles into the lumen of the stomach, lumen of the parietal cell. Likewise, histamine. Histamine, it will activate the GS subunit. Okay, it will activate the GS subunit. So it will, when it is activated, adenylyl cyclase is activated. Okay, adenylyl cyclase is activated. When adenylyl cyclase is activated, there will be formation of the cyclic AMP. When cyclic AMP is formed, protein kinase A is formed. So this protein kinase A also have action on the it also have the action on fusion of the tubular vesicles into the lumen. Right? So these three are stimulatory. Right? Gastrin, acetylcholine, and histamine. These three are stimulatory protein that stimulatory not protein transmitters, stimulatory chemicals that will cause fusion of the tubular vesicles into the lumen of the parietal cell. Okay, among these three, gastrin, acetylcholine, and histamine, among these three, histamine have the most potent effect. Right? Just remember this, histamine have the most potent effect on secretion of HCl from the parietal cell. Histamine have the most potent effect on secretion of HCl from the parietal cell. Histamine have the most potent effect on secretion of HCl from the parietal cell. Right? Just remember this. Though all of them causes secretion of the HCl from the parietal cell. Okay, this is the parietal cell. Right? Histamine secreting cell is another cell that is not present here. Right? This is the parietal cell, and the parietal cell have receptor for these transmitters. These transmitters are secreted from other cells. Gastrin is secreted from the G cells, which is present mainly in the pyloric part. Histamine secreting cells are present just adjacent to the parietal cell, nearby to the parietal cells, right? That acts in the paracrine fashion. Histamine secreting cells, they act in the paracrine fashion. And acetylcholine, it is released from the vagus nerve. Okay, acetylcholine, it is released from the vagus nerve. So these three are stimulatory to the secretion of the HCl from the parietal cell. Gastrin, acetylcholine, and histamine, they are stimulatory to the secretion of HCl from the parietal cell. Then there are substance. If HCl is secreted in higher amount, okay, if HCl are secreted in high amount, if there is maximum acidity, then the acid itself, okay, hydrogen ion itself will stimulate the somatostatin cells. Okay, it will stimulate the somatostatin cells. So when somatostatin cells are stimulated, somatostatin D cells, okay, somatostatin, they are the D cells. D cells, whenever they are stimulated, D cell will secrete the somatostatin. Somatostatin, it will come and it will bind to the receptor present on the parietal cell. Okay, D cell is present somewhere else. So D cell, it will act on the receptor present on the parietal cell. Okay, D cell will secrete somatostatin. Somatostatin will act on the receptor present on the parietal cells, and when it acts on the parietal cell, it will activate the GI. Okay, it will activate the GI. So when GI is activated, it will cause inhibition of the adenylyl cyclase. Okay, it will cause inhibition of the adenylyl cyclase. When adenylyl cyclase is inhibited, then the tubular vesicles, they will not be fusing to the lumen of the parietal cell. Okay, likewise, prostaglandin also inhibits the adenylyl cyclase enzyme. So these two are inhibitory. Somatostatin and prostaglandins are inhibitory to the parietal cells, and histamine, acetylcholine, and gastrin, they are stimulatory to the parietal cells. So this is the resting. Okay, this is the resting. These are the tubular vesicles. They are present inside the cell. So whenever they are active, the tubular vesicles, they will be fusing. Okay, tubular vesicles will be fusing, and this is the canaliculus. So in the canaliculus there is the formation of HCl. Okay, in the canaliculus there is the formation of HCl. Then that hydrochloric acid is later on secreted into the lumen of the stomach. Okay, first it is secreted into the lumen of the parietal cell or the canaliculus of the parietal cell. Then after consumption of meal they will be secreted into the lumen of the stomach. Right? So this is also the same. Acetylcholine, it acts through the M3 receptor, phospholipase C is activated. Phospholipase C causes the formation of the it converts the phosphatidyl inositol diphosphate into IP3 and DAG. IP3, it causes the influx of the it activates the endoplasmic reticulum. Endoplasmic reticulum, it causes the release of the calcium. Calcium, it causes the fusion of the tubular vesicles into the lumen of the parietal cell. Right? Gastrin, it acts to the CCK-B receptor or CCK2 receptor, you can say. Right? You should know about the receptor, right? So it acts to the CCK-B receptor. It activates the phospholipase C. Phosphatidyl, it is phosphatidyl. Phospholipase C. It activates the phosphatidyl inositol diphosphate and converts into the inositol triphosphate and diacylglycerol. IP3, it causes the release of calcium from the endoplasmic reticulum. Calcium causes the fusion of the tubular vesicles into the lumen of the lumen of the parietal cell. Right? Then somatostatin, not somatostatin, histamine, it acts on the S2 receptors. So GS is stimulated. Okay, adenylyl cyclase enzyme is activated. Okay, this adenylyl cyclase enzyme is activated, which converts ATP into CAMP. So CAMP, it activates the protein kinase A. So when protein kinase A is activated, it becomes phosphorylated for the longer time. Okay, PKA, it causes phosphorylation of the tubular vesicles. When tubular vesicles becomes phosphorylated, they will be fusing to the lumen of the parietal cell, right? And somatostatin and prostaglandin, they will cause the activation of GI, right? They will cause the activation of the G protein inhibitory. So when this is activated, it will inhibit the adenylyl cyclase, right? It will inhibit the adenylyl cyclase. When it becomes inhibited, then there is no formation of the cyclic AMP, there is no formation of the protein kinase A, and there is no fusion of the vesicles into the lumen, right? So omeprazole, it inhibits the hydrogen potassium ATPase, right? Hydrogen potassium ATPase. Atropine, it inhibits the vagus nerve. It acts on the M3 receptor. Atropine, it acts on the M3 receptor. Cimetidine, ranitidine, it acts on the S2 receptor, right? It acts on the S2 receptor, right? So these two, they are GQ stimulatory. GQ, they are S2 receptor, histamine, acetylcholine and gastrin, they are GQ stimulatory. Histamine, it is GS stimulatory. Somatostatin and prostaglandin, they are GI inhibitory. Right? Am I clear about this mechanism? So these are again the cells, right? Chief cells, parietal cells, histamine secreting cells, and G cell is present in the pyloric part of the stomach, right? So the vagal efferent, they release acetylcholine here, acetylcholine in the chief cell, acetylcholine in the parietal cell, acetylcholine in the enterochromaffin cell. Okay, vagal efferent secretes acetylcholine in the chief cell, acetylcholine in the parietal cell, and acetylcholine in the enterochromaffin like cell. Okay, but vagal efferent that acts on the gastrin G cell, that acts on the G cell, they release gastrin releasing peptide. Okay, this is important for MCQs. Right? It has been asked many times. It has been asked many times. So vagal efferent that acts on the G cell secretes gastrin releasing peptide as the neurotransmitter. In this area there is GRP, gastrin releasing peptide. In other areas there are acetylcholine. Right? So this is important for MCQs. We can keep this figure, this type of figure in the sporters also. Right? In exam we can keep this figure in the sporters. So we will be making the different cells. We will be writing chief cell, parietal cell, ECL cells, and we will be writing the G cells. Then you have to answer. Okay, we will be asking the question. The vagal efferent that acts on the G cell secrete which neurotransmitter? Okay, like that we will be asking. So vagal efferent that acts on the G cell secretes gastrin releasing peptide as the neurotransmitter. Right? So this G cell, right? So when this G cell is activated, the neurotransmitter neurons, it releases the gastrin releasing peptide. Okay, gastrin releasing peptide. So when this gastrin releasing peptide really acts on the G cell, it will secrete the gastrin. Okay, so when this gastrin is secreted, gastrin comes into the blood. Okay, it comes into the blood, it will act on the enterochromatin cell, right? It will act on the parietal cell also. This enterochromatin cell also has cholecystokinin B receptor, and parietal cell also have the cholecystokinin B receptor, and it acts on those receptors. This neuron, vagal efferent neuron that acts on the parietal cell and enterochromatin cell, they secrete acetylcholine as the neurotransmitter. It secretes acetylcholine as the neurotransmitter. Acetylcholine, it acts on the M3 receptor in the parietal cell here. Okay, it acts on the M3 receptor on the parietal cell. Histamine, it acts on the H2 receptor on the parietal cell. Histamine, it acts on the H2 receptor on the parietal cell. Gastrin, it acts on the CCKB receptor on the parietal cell and CCKB receptor on the enterochromatin cell. Okay. Am I clear? Am I clear or not? Should I repeat this again?
Yes, sir. Yes, sir. Clear, sir.
Now you can tell the mechanism of HCl secretion. Bhanna sakincha? Can you tell? Bola na. Koi ta bola.
Yes, sir. Yes, sir.
Should I repeat it again?
Yes, sir.
So this is the G cell, right? This is the G cell. G cell is present in the mainly in the pyloric part of the stomach, right? This ECL cell and parietal cell, it is present in the where? It is present in the oxyntic glands. Okay, ECL cells and the parietal cells, they are present in the oxyntic glands. G cell is present in the pyloric part, pyloric glands. Okay, so the neuron, this is the these cells, they are stimulated by the vagus nerve. Yes or no? These are stimulated by the vagus nerve. Vagus nerve is the parasympathetic nerve. Right? Vagus nerve is the parasympathetic nerve. Whenever there is parasympathetic activation, there is increase in secretion from the GI tract. Yes or no? Right? Whenever there is parasympathetic stimulation, there is increase in secretion from the GI tract. Right? Secretion is increased. So this vagus nerve is acting on the parietal cell and it is acting on the enterochromatin cell by releasing the acetylcholine. So when it acts, when this acts, so acetylcholine, it acts on the M3 receptor on the parietal cell. So when acetylcholine acts on the M3 receptor on the parietal cell, what happens? There is the increase in calcium level, right? There is the increase in calcium level. So when calcium level is increased, the tubular vesicles, they are present inside the parietal cells. They will be fusing to the lumen of the parietal cell, fusing towards the luminal side of the parietal cell, and HCl is secreted. The same vagus nerve that acts on the enterochromatin cells, they will cause release of the histamine. They will cause release of the histamine. Histamine, it will act on the H2 receptor, histamine 2 receptors. So when histamine 2 receptors are activated, it acts on the parietal cell, it will cause formation of cyclic AMP and ultimately protein kinase A. So when protein kinase A is formed, it becomes phosphorylated and when it becomes phosphorylated, the tubular vesicles consisting of hydrogen potassium ATPase that will be fusing towards the lumen of the parietal cell, and hydrochloric acid is secreted. Likewise, gastrin, okay, gastrin, it is supplied by the vagus nerve itself. Okay, it is also supplied by the vagus nerve, but the cells that innervate the G cells, okay, the neurons that innervate the G cell, they do not release acetylcholine. They release the gastrin releasing peptide as the neurotransmitter. Gastrin releasing peptide causes release of the gastrin, okay, release of the gastrin. So gastrin will be released, it will be going to the circulation. Okay, remember this, it is not acting through the paracrine fashion, right? Histamine, it is acting through the paracrine fashion, it is not going in the blood, but gastrin, it is going into the blood through the circulation, it reaches to the ECL cells and it reaches to the parietal cells. It acts on the cholecystokinin B receptor on the ECL cells, and it acts on the cholecystokinin B receptor on the parietal cells. And after going inside, it will cause activation of IP3, inositol triphosphate is increased. It will cause activation of inositol triphosphate, and it will cause increase in calcium level. When calcium level is increased, calcium causes the exocytosis of the tubular vesicles. It causes the fusion of the tubular vesicles consisting the hydrogen potassium pump. Okay, tubular vesicles consist of the hydrogen potassium pump that will be fused to the luminal side, and there is the exocytosis of the transmitters, right? And there is the exocytosis of the exocytosis, right? Am I clear now? Right? Clear?
Yes, sir.
So अर्को भयो D cells, right? D cells, when hydrogen ion is in excess amount, right? Hydrogen ion अब धेरै भयो, when hydrogen ion is in excess amount, then this hydrogen ion, it will activate the D cells. So when D cells is the somatostatin cells, okay, so when D cells is activated, D cell, it will secrete the somatostatin, it will inhibit the GS subunit. Okay, it will inhibit the adenylyl cyclase enzyme. So when adenylyl cyclase enzyme is inhibited, then these will not be secreting the gastrin. Okay, this will not be secreting the gastrin into the circulation. Right? So this is also the same. Right? Vagus nerve here. Here is the gastrin secreting cell in the pylorus part. Right? Gastrin secreting cell is in the pylorus part. Other cells are present in the fundus and body. Okay, fundus and body part tira huncha. Aru gastric glands are present in numerous in the fundus and the body. Right? So vagus nerve, it releases acetylcholine here. Okay. Vagus nerve, it releases gastrin releasing peptide here. Gastrin, it goes through the blood, it acts on the parietal cell, and there will be secretion of HCl and HCl. Okay, if HCl is more, then gastrin is also inhibited. Okay, gastrins, G cells is high pH will also stimulate the somatostatin cell. Okay, if the lumen of the stomach becomes more acidic, D cell will be stimulated as well as G cell will be inhibited. Right? So this is also the same. Right? This is the D cell, this is the H cell, this is the parietal cell. In the parietal cell, there is the M3 receptor. In that, the vagus nerve will release the acetylcholine as the neurotransmitter in the parietal cell, and it will release the acetylcholine as the neurotransmitter in enterochromaffin-like cells or H cells. So they will, when it acts on the parietal cell, calcium level is increased, there is the secretion. When it acts on the histamine enterochromaffin-like cells, it acts on the H2 receptor, histamine is released. When histamine is released, it will cause secretion of the HCl. Same vagus nerve, it acts on the D cell by releasing the gastrin releasing peptide. Okay, it goes through the blood and it will stimulate the H cell. Okay, there is the CCK-B receptor. This gastrin, it will go through the blood. Okay, gastrin, it will travel through the blood, and this gastrin, it will go to the CCK-B receptor. It will act on the CCK-B receptor and it will cause secretion. It will cause increase in calcium level in the parietal cell and it will cause secretion of the hydrogen and chloride. Right? So this much for today. That is the same thing. Right? Am I clear? कतिजना छ क्लियर, कतिजना छ अनक्लियर, कतिजना छ? If I am unclear,
Yes, sir.
You,
Yeah.
sir, post something alkaline tide के हो spelling?
a postprandial alkaline tide, right? Postprandial alkaline tide means when you consume meal, after consumption of the meal, the HCl is secreted. Okay, HCl is secreted. When HCl is secreted, the chloride ion, it comes to the lumen of the stomach. So for compensating that, there is the exchange of bicarbonate from the parietal cell into the blood. Okay, so when bicarbonate ion goes into the blood, it increases the pH of the blood. Okay, it increases the pH of the blood, which is known as postprandial alkaline tide. Right? Am I clear? So may I leave?
Thank
So much.
you, sir.